Once the desired edits have been made to the genes need to be screened in order to see whether or not they have the required gene. Two methods can occur. The first is PCR and the second is FISH method. PCR stands for polymerase chain reaction, and it is a powerful process used to make millions of copies of a DNA sequence without the host organism. Once the copies of the target gene have been amplified, they can easily be examined to see whether or not they carry the desired gene. This is a three-step process, and the first step is denaturation. The DNA is denatured into two strands, and it is done so by being heated up to 96 degrees for 20 to 40 seconds. The second step is annealing. This also lasts 20 to 40 seconds, but it is done at a different temperature. It is done at 50 to 65 degrees Celsius. DNA primers anneal to each of the strands, and two primers are used, one that goes forward and one that is reversed, as one is complementary to a sequence near the 3' end and the other anneals to the sequence near the 3' end on the other side. And both of them move in a 5' to 3' direction towards the target sequence. The third step is elongation. This is done at 72 degrees Celsius, and the original strands act as templates as DNA primases add more and more nucleates until the entire gene is done. At the end of the first cycle, there are now 4 strands, as we originally began with 2 and those doubled. So the second cycle is now run with 4 strands. And then the third cycle is run with 8 strands, and by the end of the third cycle, there are 2 matched target molecules. And as the process goes on, the gene is amplified, and scientists can easily isolate the gene and study its sequences. ISH stands for fluorescent in situ hybridization. It is an extremely useful method that allows scientists to discern the location of genes on a chromosome. In terms of our research, it would allow the scientists to see whether or not the desired gene has been added effectively. The first step is to create a hybridization probe that is complementary to the target gene. It is important that the probe is labeled with a fluorescent marker. The chromosomes are then put into a slide and denatured, and then the denatured probe is added to the same slide, and the two are allowed to hybridize. When the slide is observed underneath a fluorescent microscope, it will show the places that the probe has hybridized to, and we can see exactly where those genes are on the chromosome. The chain termination method helps us understand how genes work by mapping out the nucleotide sequence. 4 reaction tubes are set up with the nucleotides, single-stranded DNA, DNA primers, DNA polymerase, and ddNTPs. All 4 tubes get all of the materials, except each tube only gets one type of ddNP, either ddATP, ddCTP, ddGTP, or ddTTP. The ddNTPs are important because when they bind with their complementary nucleotide, nothing else can bind after them due to their lack of an OH group on carbon 3. This terminates the sequence, and what we are left with is different strands of different sizes. We can then see what the last nucleotide is using either fluorescence or gel electrophoresis.